Hey guys, today we're going to focus on genomes and genomics, so technology that we use to analyze genomes themselves. So first, the question we should ask is, what is a genome? Genome is the complete set of genes or genetic material present in a cell or an organism. And genomics, that is the study of a full genetic complement of an organism or the genome itself, including interactions with the different environmental factors that exist. So there are a couple of types where it's like a different parts of genomics, where the first one is comparative genomics. This part of genomics, it aims to compare genomic features and differences between species. The next one would be structural genomics. Structural genomics involve the aim to determine the structure of every protein encoded by the genome. Then we have functional genomics. Functional genomics aims to collect and use data from sequencing for describing these genes and various protein functions. I will fix that typo afterwards. So in this talk, we'll have a couple of things going on where we keep saying cDNA or genomic DNA. When we talk genomic DNA, we should think unprocessed DNA, and it is the DNA before any transcription or translation or any other fun gene expression methods occur. Whereas cDNA or complementary DNA is the DNA that is synthesized from the mRNA template. So that cDNA is actually coming out of the mRNA reverse transcription of the mRNA that we have. Okay. So one of the methods you would use to analyze genomes is called whole genome sequencing, or this is also called shotgun cloning. It is a widely used strategy for sequencing and assembling an entire genome. So it kind of happens in 3 stages of sequencing and kind of getting everything together. So first, the DNA is cut into fragments, so it can be different restriction enzymes and all that fun stuff that cleaves DNA at certain spots. It's contigs there that end up and it forms contigs, which are just continuous fragments of the DNA itself. The entire chromosome is then assembled and by a computer program, and then the fragments are aligned based on identical sequences depending on the pattern of how they are cut and put together. So what is the difference between a contig and a scaffold? Scaffold is, so contigs. We know that contigs are sequences of overlapping reads that are assembled into units. So we have certain sequences that overlap and they are separately assembled. Whereas scaffolds, they're a collection of contigs, right? So contig 1, contig 2, they come together, they become a scaffold. This is why they're also called supercontigs, because they have many, they can have two or more contigs joining together. So it kind of ends up getting called supercontig. Then, so the logic behind obtaining a genome sequence kind of revolves around these 4 steps. First, you cut it all into mini fragments, because we have DNA genomes that are huge, right? So we want to break it down into small pieces, just like how we study things, right? We break everything down. So to study the genome sequence itself, we need to break them down into different fragments. So that's the first part. Then we sequence them, like, for example, like Sanger sequencing, or in this case, whole genome sequencing techniques to sequence each and every fragment. Then you take those sequence reads and you overlap them. So remember how we did like gene mapping based with it overlapping the different genes and all that? It's a similar thing here where they kind of just overlap based on continuous ends, right? So they come in, oh, like, oh, so this is here, this matches up here, so we're going to add that there and then go there. This is kind of the same thing, just depending on where it's being overlapped and all that fun stuff, right? So we overlap these sequences, and then these overlap contigs, which end up our scaffold, right? So that the scaffold is essentially comes in, the contigs end up overlapping with each other, and then you put them together to find that whole sequence. So why must the DNA fragments be sequence overlap in order to obtain a genome sequence? So essentially, this question is asking, why do these DNA fragments have to overlap in order to get that genome sequence? Because it's required to align the sequences relative to each other, right? So when you have similar things going on, it allows you to, the overlaps actually allow you to see which one is next to each other or not, okay? Oh, okay. So filling in the gaps in draft genome sequences is a major challenge. Based on figure 14.6 in the textbook, can paired end reads from a library of 2 kb fragments fill a 10 kb gap? So we have this figure here. They're asking if paired end reads can of from a library of 2 kb fragments fill this gap that is 10 kilobases long. And the answer is no, because typically these gaps consist of repetitive sequences, and sequencing reads in these regions cannot be aligned with certainty. So essentially, they cannot align with each other, since typically these gaps end up with repetitive sequences. So essentially, you cannot build it back together without any certainty. So that is why these gaps come up, because the sequences in the region of the gap cannot be easily cloned in E. coli and would therefore not be represented in the libraries. So these gaps come up because they cannot be aligned with certainty. In this case, since it cannot be easily cloned, and it wouldn't be represented in the libraries that you would use for these different situations, right? I hope that made sense. In figure 1410 from the textbook, express sequence tags are aligned with genomic sequence. How are ESTs helpful in genome annotation? I'm sorry, guys, I forgot to put my animation in a couple of these slides. But anyways, this is the figure 1410 out of our textbook. It's asking how are these tags helpful when you are doing genome annotation? This is because ESTs, or express sequence tags, they actually help sort out which are the 5' ends and which are the 3' ends of that transcription unit. So once you know the 5' and 3' ends of a transcription unit, you know that these exons that we need to transcribe actually are within that region, right? Within those boundaries of 5' EST and 3' EST. So your exons are right in between. Explain the approach that you would use to apply to sequencing the genome of a newly discovered bacterial species. So in this case, you would definitely use whole genome shotgun approach because bacteria have relatively small genome, essentially roughly around 3 megabase pairs. And there are typically no repeating sequences. Since it's small enough, they don't need that much repeating sequences to keep it settled down. So in this case, you would just use the whole genome shotgun approach because you would essentially the size itself can determine, whereas it would take longer with human genomes, which are even bigger than a small little bacteria, right? Okay. In 14, and so that same figure we saw earlier, complementary DNA sequences are aligned with genomic sequences. How are cDNA sequences helpful in genome annotation? Are cDNAs more important for bacterial or eukaryotic genome annotations? This is essentially because cDNA defines the exons of a particular gene, and the sequences in between essentially represent the introns. So as a result, complementary DNA are more important for eukaryotic genome annotation because the bacterial genes end up lacking because it lacks the introns itself, right? So next we have what is called the Human Genome Project. The Human Genome Project is an internationally coordinated effort to sequence and identify all genes of the human genome. This illustrated that humans and other species share a common set of genes that are essential for cellular function and reproduction. And this project actually fueled the discovery for more than 1,800 disease genes. So this is how we kind of come into functional genomics, because we kind of figured out how many of these genes, and the project itself, it kind of gave rise to what is called functional genomics. Functional genomics is the study of gene functions based on resulting RNAs and proteins they encode. So it's trying to understand different genes and how they work, essentially. We can predict functions using different BLAST search, which is BLAST is essentially a database, and it contains a lot more databases of genomes and genomic data and sequences. So through a BLAST search, so essentially we can use NCBI. BLAST kind of goes into searching different genome sequences, right? So you essentially take that, you screen these different databases with a sequence, and you compare that sequence to a known sequence that is already in the system of that database that they're looking in. Then we have a search that is called similarity searches, and those are essentially trying to see how similar a genome sequence is statistically with a known function that is likely encoded for a protein with a similar function. Annotation in this case kind of relies heavily on bioinformatics, and the process of identifying allows the process of identifying genes, the regulatory sequences, and their functions. Transcriptome analysis is essentially a high throughput analysis of all expressed genes that can be conducted by two main approaches. One way to do this is sequence-based approaches like RNA-seq. That's just one example. There are multiple other ones. And we have microarray-based approaches. So here's a question that just goes on the human genome itself. The human genome contains approximately 20,000 protein coding genes, yet it has the capacity to produce several 100,000 gene products. What can account for the vast difference in gene number and product number? Essentially explain what you think your answer, so justifying your answer. So essentially we know that many genes encode for the multiple proteins through alternative splicing, right? And alternative splicing patterns, as we've seen before, can generate multiple mRNA molecules, and with the different mRNA molecules, you can actually end up with different protein molecules, right? Okay, so this is another question. You have sequenced the genome of the bacterium Salmonella typhimurium. You are using a blast analysis to identify similarities within the S. typhimurium, right, to known proteins. You find a protein that is 100% identical in the bacterium E. coli. So Escherichia E. coli is E. coli. When you compare nucleotide sequences of the Salmonella typhimurium and Escherichia E. coli genes, you find that their nucleotide sequences are only 87% identical. Explain this observation. So why do you guys think that this is 87% identical? Because as we remember, our genetic code itself, or the triplet code that we use, is redundant, and any changes in the DNA nucleotide can occur without changing that sequence for the protein itself, right? So it's not changing the sequence of that amino acid. So this is a second part of that question itself. What do these observations tell you about the merits of nucleotide versus protein similarity searches in identifying related genes? So protein sequences are expected to evolve and diverge more slowly than the genes themselves, because the flexibility of the third positions of most codons. So remember how in some of the codons that we looked at with the genetic codon table, we saw that some of them with the third position different had the exact same amino acid coded for it, right? So that just tells us that it's so flexible. With that flexibility, the DNA sequence can accumulate changes without actually changing that protein itself, right? Because the amino acid is essentially staying the same. The amino acid changes may change, as I said, so this change is do not affect the protein structure itself, this because it's not making any changes to the amino acid itself, right? So if amino acids end up changing, that is what changes the function of structure and function of the proteins, right? Because remember, with the translation and protein talk we had, we know that the amino acid sequence of that protein is what determines the structure, right? And the structure itself determines the function, right? So since the amino acid sequence is functionally constrained, natural selection will eliminate any deleterious amino acid changes. So essentially with that, so if any changes in amino acid sequence occurs, that essentially gets eliminated by natural selections, because any issues that come in, they kind of just like, oh, nope, does not work. Bye bye. No, not today. So essentially, this changes the rate of change in the amino acid sequence and leads to a greater sequence conservation of amino acid sequences compared to the nucleotide sequences. Because like even if the nucleotide changes, you can still do changes within that. Okay. So but when the amino acid ends up changing things, they end up changing everything that they need to do. So there's more flexibility with DNA changes than amino acid changes in this case. Okay. So that's what these observations are telling you, in depending on how similar these guys would be and how different, even though we have different DNA sequences, we can still have similar proteins come out. Okay. So in this case, how are the positions of the codons determined? Several methods. So this is figure 14.9. So to there are several methods that involve the sequences of proteins. One of the ways is like computer analysis of the nucleotide sequence to look for open reading frames that can that can identify at least some of the exons. So this is it starts off trying to find the exons, and these searches end up getting refined to actually detect codon bias, binding site predictions, and conservation of predicted amino acid sequences. So it kind of brings down what is happening, like, ooh, yay, we kind of get what's happening here, right? So you kind of come in to be like, okay, so this is around the area where it started, right? So if you have like a methionine here, it's like, oh, that's where we started. Woohoo, yeah. And then we can have like a terminator codon somewhere here, and it's like, oh, it terminated. So we know that there is a termination site at this area here. Okay. So that kind of comes down to how it kind of gets determined. Then there are other situations where we can use comparison of the complementary DNA sequences to the genomic sequence and directly reveal how those sequences contain these coding portions of the gene, right? So that is essentially how you can determine the position of the codons themselves. So DNA microarray analysis. This is essentially so a method to do this is using microarrays, or called it's called gene chips, like this guy. So this is where single-stranded DNA molecules attached to a glass microscope slide. So it kind of comes in here, it attaches to this glass area here, and then it ends up through a system, it gets in here, and then it comes into this, it scans everything in, and this guy is called an arrayer, and it gives you different gene expression levels. And this is typically used by cluster analysis groups genes. So these little dots over here are kind of called clusters, and it kind of shows how gene expression is upregulated, or where it is upregulated or downregulated. So for microarray analysis, cDNA, complementary DNA is used to determine levels of gene expression. Explain how a microarray is used to evaluate gene expression levels. So in this case, the experimental and control complementary DNA are tagged with different fluorophores. The complementary DNA is then hybridized to the microarray chip with a reference genome, and the fluorescence is analyzed for each gene. So this is an example where gene expression is compared at normal temp versus high temp, right? So you see different colors here, right? So when you see that, when you see black guys, black no fluorescence or black colored areas, you see that there is little or no gene expression. When you see green, there is downregulation of that happening. Red shows very much high regulation or upregulation of that. But then when you see the yellows, those show no change in gene expression. So in this scenario, they have it like, so in this case, they had high green spot. When they had green spots, the genes were transcribed at normal temperature. So in this case, in normal temperature, the gene itself was downregulated. In yellow spots, it was equal within both cells. So in normal temp and high temp, it kind of got, it was on the, it was, there was no change between the cells. And then with dark spots, there was low gene expression in general. And then here we see that with the high temperature ones, the red ones ended up having upregulation of the gene itself at the high temperatures. So I hope that does make sense with microarrays. What is the difference between forward and reverse genetics? So forward and reverse genetics are essentially two different types of analyzing genes and how they kind of work. So forward genetics is essentially the approach of determining the genetic basis for that is responsible for a phenotype. Whereas reverse genetics is trying to understand the function of a gene by analyzing the phenotypic effects of a specifically engineered gene sequence. Forward genetics, you start from the phenotype. Reverse genetics start from the gene sequence. And then forward genetics is involved with identification of the gene responsible for particular phenotypes. Reverse genetics is involved in the function or possible phenotype of an altered gene. So essentially with this one, you kind of start with random mutations, whereas you would do mutations in reverse genetics that kind of disrupt that gene itself, like to see how that functions or what phenotypes they get to understand how this gene actually works. I hope that makes sense. Gene targeting. So gene targeting is a technique that uses homologous recombination to modify an endogenous gene. It is used to inactivate or modify a functional endogenous gene and allows us to study gene function and expression itself. Some methods that we use for this is like knockouts, where it completely disrupts the expression of specific genetic loci. Knock-ins are typically when a desired gene or sequence is inserted into a specific locus in a target genome. So, okay, so this is a question. It's a fun one. I really like it. When generating genetically modified mice using homologous recombination, chimeric mice are often generated. Explain what chimerism is and why this occurs in this process. So chimerism, it's when a single organism contains two different sets of genomes. When generating genetically modified mice, recombinant cells are actually injected into wild type mice blastocyst, where they mix and form a mosaic cell mass. So essentially, when we have like these guys, you see how the differences come in. Be like, ooh, whoa, we have green mice happening. What? Okay, so this is all I have for you guys today. If you guys have any questions, don't hesitate to contact me. See you guys.